Carbohydrate in food. The course outline for this module starts with the introduction, classification of carbohydrate based on the sources and structures, physicochemical and functional properties of different classes of carbohydrate. First topic is introduction. So basically, carbohydrates are easily found anywhere in the nature, including in your body, outside your body, and anywhere you look, you will find a lot of things or creatures that are made from carbohydrates. The glucose is the source of energy for plants and for human. The energy reserved for plants are starch, and the energy reserved for human is glycogen. It also becomes the structural material for plants and insects, for example cellulose, xylans, and mannans. Chitin is the structural material for insects, so if you look at insect that has a hard exterior texture on the outside of their body, and let's say if you step on them and there's like a crunch, so that means it has a chitin cell wall. Carbohydrate can also become a protective substance for plants in the seed coat or the cell walls of the endosperm of a cereal. Carbohydrates can also become a precursor for other biomolecules and derivatives, for example DNA, RNA, glycoprotein, and glycolipid. There's a lot of carbohydrates function in food. First, it provides some taste. If you talk about simple sugars, definitely you will have that sweetness, but then if you talk about complex carbohydrates, normally they don't have that sweetness. So it depends on types of carbohydrates that you are consuming. Carbohydrate gives that structure or texture of food. For example, we have starch in bread pudding. The higher the amount of starch, the thicker the concentration of pudding can be. We also have dextrin that is actually very good as a coating or batter texture, and pectin that gives the gelling properties of jellies and jams. So those can also help with the texture properties of food. Carbohydrate also helps to lower the water activity of food and influence ice crystallization in ice cream. It can also be a fat substitute, especially for diet food. For examples, modified starches and also hydrocolloids. It also gives the desirable flavor and color in food that is caused by a Maillard browning and caramelization. Carbohydrate is also good fiber source depending on the type of carbohydrate. For this type of carbohydrate, we're talking about complex carbohydrates and not the simple sugars. Some of them have dietary fiber, resistant starch, or slowly digestible starch depending on the type of carbohydrate and the structure of the carbohydrate itself. Basic structure of carbohydrate composed of the elements carbon, hydrogen, and oxygen. It consists of simple sugars, and it also contains alcohol and carbonyl functional groups such as aldehydes and ketones. Not all carbohydrates have this empirical formula of carbon, hydrogen, and oxygen, for example deoxy sugars and amino sugars. They don't actually comply to this empirical formula. So for carbohydrates, even though they have the same molecular weight, but then the rotational functions of the carbohydrates and the large number of the asymmetric can give different kind of properties or features. Moving on to the classification of carbohydrate. Classification of carbohydrates can be divided into two. Simple carbohydrates, for example ketoses, aldoses, and any compounds that can be derived from them via a few processes like condensation, hydrolysis, and also reduction. Next is complex carbohydrates, for example cellulose, polysaccharides, glycoproteins, and proteoglycans. Basically, carbohydrates can be divided into four basic groups. Monosaccharide is the simplest form of carbohydrate because it has only one basic structure of glucose. Disaccharide has two monosaccharides bind together. Oligosaccharide has the basic compounds of glucose up to 10 units. And finally, polysaccharides with which have more than 10 monosaccharide units, so they are categorized as complex carbohydrates. Moving on to the subtopic of monosaccharide. Sources of monosaccharide. Glucose and fructose can be found in honey, molasses, dried fruit, and fruit. Galactose can be found in milk. Carbohydrates contain chiral carbon atoms. A chiral carbon atom is one that can exist in two different spatial arrangements. Chiral carbon atoms have four different groups attached to them. The two different arrangements of the four groups in space are non-superimposable mirror images of each other. Structure of monosaccharide. One of the carbon atom has a double bond to an oxygen, so this is called carbonyl group, and then it is called so-called an aldehyde. The other carbons are attached to a hydroxyl group or an alcohol, so they are called as hydroxyl group or alcohol. It is considered as polyhydroxyaldehydes or polyhydroxyketones because they contain several hydroxyl groups in either an aldehyde or ketone group. Aldose means aldehyde sugar, while ketose means ketone sugar. Monosaccharides can also be classified based on the number of carbon atoms. For example, if it's only 3 carbon, it is triose sugar. If it's 5, then it will be pentose sugar, and if it's 6, it is called hexose sugars. There are different kind of projections of monosaccharides. On the left is wedge and dash with example of D-glucose. Pay attention to how it is being numbered. So the carbonyl group normally would be the number 1 carbon, and then the next one would be the next numbers until the final number of carbon. In dash projection, wedges indicates chemical bonds projecting outwards from the plane of the page. For Fisher projection, it is much simpler. Everything is just in a straight line like that. For monosaccharides, there's also enantiomers. Basically, it is a mirror image, for example, D-glucose and L-glucose. The difference between these one is the positioning of the hydroxyl group at the 5th carbon. Just imagine that there's actually a mirror in the middle, so it's just a mirror image, but they have different chemical and physical properties. Structure of carbohydrate can be found in linear form and cyclic form. Pay attention again to the numbering of the carbon. The cyclic form is actually much more stable as in the linear form. To explain the formation of cyclic form, the alcohol group will attack the aldehyde group to form the ring, the cyclic. Basically, the hydroxyl group from the 5th carbon will attack the oxygen in the carbonyl group of all the aldehyde group. So, 2 different kinds of stereoisomers that can actually form: alpha enomer, which is the basis of starch, and beta enomer, the basis of cellulose. The difference is only the positioning of the hydroxyl group. For the alpha enomer, it's also going out downwards, while for the beta enomer, the hydroxyl group is actually going upwards. Even though they look very similar and they have the same molecular weight, they have completely different properties. For example, alpha enomer in starch is digestible, but for beta enomer like cellulose is not digestible in our body. Cellulose and considered as fiber. The alpha and beta forms interconvert through a ring opening transformation reaction called mutarotation. There are about 36% alpha D-glucose and 64% beta D-glucose in an aqueous solution of D-glucose. The ring structure can be found as 6-membered ring structure, which is called pyranoses such as glucose, and 5-membered ring structure, which is called furanoses such as fructose. Moving on to the next subtopic of disaccharides. Sources of disaccharide. The 3 main disaccharides are sucrose, maltose, and lactose. Sucrose forms by glucose and fructose, can be found in table sugar, sugarcane, and sugar beet. Maltose forms by 2 glucose units and found in malted or germinated cereals and legumes. Lactose forms by glucose, and galactose is mainly found in milk. Disaccharides are formed from 2 units of monosaccharides. Once they are formed, it will release water and will form a glycosidic bond. These are the examples of formation of disaccharides. Focus on the numbering of the carbons to know how the linkage are actually being named. For maltose, the linkage is named as alpha-14 linkage because the hydroxyl group going downward. It is actually alpha, and these 2 glucose and glucose are connected at carbon number 1 and 4. So that's why it is called alpha-1, 4 linkage. The next example is lactose. D-galactose combined with glucose, and the linkage is beta-1, 4 because the hydroxyl group before they are linked it is going upwards, and the galactose and glucose are linked at carbon number 1 and carbon number 4. So that's why it is called as beta-1, 4 linkage. Now let's have a look at different types of disaccharides that is available. Maltose is a malt sugar and formed from the breakdown of starch. An example of food that contains maltose is malted barley, where they are being germinated so the starch will be converted into maltose. So normally malted barley is used in the brewery process where they produce alcohol. Maltose is also a reducing sugar, so we're going to talk what is actually reducing sugar more after this. The next one is lactose. Lactose is normally found in mammalian milk. To those that does not have lactase, which is an enzyme that breaks down lactose into 2 monosaccharides units, they are called lactose intolerant. What happened is when these lactose are not digested in the stomach, the intestinal bacteria in the large intestine are the one that's going to break it down, and they'll produce a lot of gases, which will cause cramping of your stomach. It is also a reducing sugar. The next one is sucrose, known as a table sugar that we use it a lot in our kitchen. It is the most abundant disaccharide in nature and found in sugarcane and sugar beets. Since it has no free reducing end, it is not a reducing sugar. So how could you identify which is a reducing sugar and which is not a reducing sugar? The definition of reducing sugar is sugar that contain free aldehyde groups or keton groups that act as a reducing agent, and they're oxidized to become carboxylic acids. So the reducing capability is defined as the presence of free or potential aldehyde or ketone groups. Remember that aldose or ketose are actually not stable, so they can become as hemiacetal to form an intermediate. To explain further, let's have a look at some examples. Mannose, galactose, and fructose have hemiacetal ring, and in the nature, they can form into an open-chain aldehyde. The hydrogen will move to this oxygen and it will be open-chain aldehyde, so whenever this happens, then this structure will become a reducing agent. It can react with other kind of structure to become much more stable. So this is an example of lactose, which the acetyl is very stable because the hydroxyl groups from these two units are attached together. The hemiacetal group has the hydroxyl group that can actually attack and be an open-chain aldehyde. Right, same thing with lactose. So over here, if you look at sucrose, it has acetyl groups that are very stable and unable to form into open-chain aldehyde. Then it is not a reducing sugar and it cannot be a reducing agent. This is an example of a reducing sugar test, namely a Benedict test. The monosaccharide reacts with copper, which is actually a blue solution, and then the oxidation of the deep glucose will form the gluconic acid and a brick red solid. The copper is being reduced by the aldehyde in the glucose, so if it is positive, then it will become a red solution. But if it's negative, then it maintains its blue color. Reducing sugars act as mild reducing agents or electron donors and catalyze desirable browning of food in the presence of amino acids. The reducing property is as a result of the presence of the reactive carbonyl group in the open chain form, but not in the ring form of reducing sugars. When the few open chain molecules are removed from solution by the reaction, a number of the ring molecules open to replace them in order to re-establish the original proportions present. Moving on to the physicochemical and functional properties of sugar. Sweetness. Sugar contributes to 4kcal. Different sugars have different sweetness. Relative sweetness. Sucrose is used as a standard of 1.0. Hygroscopicity is the ability to attract and hold water. It is useful to maintain the freshness of some caked products. However, it is also a source of texture problem when relative humidity is high. An elevation in temperature increases the absorption of moisture from the atmosphere. Solubility. Sugar is soluble in water because of numerous hydroxyl groups. Sugars have different solubility in water. As the temperature of water increases, the amount of sugar capable of being dissolved in water increases. Lactose is the least soluble as compared to the others. Fructose has the highest water solubility that affect the crystallization, vapor pressure, freezing point, and boiling point. For example, in honey we have fructose and sucrose. If we put it in a fridge, you can see that it crystallizes, and sometimes there's like one layer at the bottom of honey that shows crystallization of sugar. Some people say that that's not an original honey when there's crystallization. Well, it crystallizes not because they add sugar inside it, but it's because of the nature of the glucose that this does not solubilize in water, and it will also affect the freezing point. So that's why if people say, oh, that's not an authentic honey, well, could be wrong. To conclude this lecture, you should know what are monosaccharide and disaccharides, their sources, classifications, physical and chemical properties. These are the references. I will see you in the next video for Carbohydrate Part 2.